Okay, now dear student, in previous lecture almost you are aware with the various concepts of DNA, RNA, nucleotides, nucleosides. So now in today's lecture we are start with what replication. But prior to start with what replication, we must recall our basic things what we have studied in previous class. During gene expression inside the cell, the DNA they are expressed to mRNA. From mRNA, synthesis of amino acids or you can say proteins takes place on what ribosomes. The process which involves synthesis of RNA from DNA, that process is called as what? Translation. Almost you are aware with this. Okay, definitions. From RNA, amino acids or proteins they are synthesized, and that process is said to be translation. Now, when the process in which from synthesis of daughter DNA from parent DNA takes place, okay, that process is called as replication. Now, in today's lecture, we will focus on replication. So, what do you mean by replication? Actually, replication is of two types, that is autocatalysis and second one is said to be heterocatalysis. During autocatalysis process, the synthesis of daughter DNA from parent DNA takes place. That means DNA to DNA is said to be what? Autocatalysis. And in heterocatalysis, from DNA, RNA is formed, RNA is synthesized. Actually, let me tell you one thing, the replication is a very complex process. This replication, it involves various enzyme machinery. The most important enzymes, let us try to understand, which participate in the replication process. The first enzyme is said to be Helices. Now let us try to understand. Note down the first enzyme, so called what? Helices. What is the role of Helices? This Helices, actually we know that DNA is a coiled molecule. This DNA structure is in this form, in coiled form. This helices is responsible for conversion of coiled structure, okay, into this type of structure. So, in brief, let me tell you one thing. This helices, it opens up the two strands of what? DNA. Here, breaking of hydrogen bonds takes place, and helices is responsible for breaking of hydrogen bonds. So this is the role of what? Helices. Okay, helices converts this coiled form of DNA to uncoiled form. The next enzyme which is responsible for replication process is said to be topoisomerase. Next enzyme is said to be topoisomerase. Now, simply you should note down about the function of topoisomerase. It avoids recoiling of open, or you can say separated DNA strands. So this is the function of topoisomerase. It is responsible, or it avoids the recoiling of open, or you can say separated strands. Next, third enzyme which participate in replication process is called as ligase. This ligase, it is responsible for sealing of gaps. or simply we can say that it is responsible for joining of two DNA fragments. Fourth important enzyme, sorry, okay, that is DNA polymerase. This DNA polymerase 1, now there are three types of DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. This DNA polymerase 1, it helps in removal of primer and filling of gap. This is the function of DNA polymerase 1. It is responsible for removal of primer and filling of gap. We will try to understand this basic phenomena afterwards as we will proceed in detail about what replication. Now the next enzyme which is associated with replication process is said to be primase. This primase is responsible for primer synthesis. Primer is nothing but it is a small fragment of RNA containing not more than, it is a small RNA fragment containing not more than 10 nucleotides. That means RNA primer always contains less than 10 number of what? Nucleotides. Now the next enzyme which is associated with DNA replication process is DNA polymerase 3. Dear students, this is the most important enzyme participating in DNA replication process. This enzyme, it is also responsible for exonuclear activities. This enzyme is also responsible for exonuclear activities. It is one of the most important key enzyme. Rest of the enzyme, they helps in replication process, but the most important enzyme considered for replication process is said to be DNA polymerase 3. Next enzyme which is associated with DNA replication process is phosphorylase. This enzyme phosphorylase, it helps in addition of phosphate this enzyme phosphorylase, it converts monophosphate nucleotides to triphosphate nucleotides. This is the most important function so far phosphorylase to be concerned. It is responsible for conversion of monophosphate nucleotides to triphosphate nucleotides. In brief, this phase is said to be the activation phase, or simply we can recall it as a activation of nucleotides. So we can also say that phosphorylase enzyme, they are responsible for activation of nucleotides.